Hi everyone. In this video, we will start with the first annotation approach, that is marker based approach. So in the last video, I talked about the foundation of single cell annotation, why we need to do that and what are the challenges we see and different assumptions. Then I discussed like there are three approaches. One is the marker based approach, the second one is the reference based approach, and the third one is very recent, the AI assisted approach. So in the marker based approach, what we do, like we take the differential expressed or the top markers for a given cluster, and we compare that against some marker databases. So in this video, we will explore different marker databases that you can use for the marker annotation method, and in the next video we will see how this approach works. Okay, so we'll focus on different marker databases that are available, which helps us to annotate the given cell type. Okay, so with that, let's start. Okay, this is what the marker based approach, like you take a cluster, you find that gene and you compare with the known markers, and for this information, for that we need some databases where they will provide this information. Okay, so let's call that marker databases. Okay, so this is a paper from 2023 where it's a review paper. They have so you will see that there are many databases that are there for the single cell annotation marker databases. Some of the most widely used of this list are DB, then CellMarker, very well known, MSigDB and Tissue, even Azimuth, those kind of things are there. And okay, the list is comprehensive, and the data sources are single cell, and some are single plus bulk, some are microarray based, and like that. Okay, and also these different sources will be different with respect to what tissue they cover, what healthy samples, disease samples, those kind of things. So that's what again, when we are downloading or referring these kind of databases, we have to carefully choose the sample that is very close to the experiment that you are doing with. Okay, so we will not cover all the databases, but few of them that I will be covering, and that will give you an idea like how this looks like. So we'll start with the first CellMarker databases, and this is the URL is is like this. Google, if you search CellMarker 2.0, it will give you some other one. This is the correct URL that you can look into. And this is a very recently they have updated it. That's a 2.0, and it contains roughly this many markers across this many cell types and 656 tissue. And their species this is for human as well as for mouse, and within human you have blood, brain and liver and other tissue types are covered, and they have pulled the information from almost this many literature paper, and the data comes from even single cell, bulk, all kind of sources they pull from. Okay, so we will explore that. Okay, yeah, this is the CellMarker databases. If you go to home page, this will look like this, and you can select a tissue and get further information. And yeah, the left hand side also, this gives some statistics, like for in case of human, how many tissue type it covered, and what kind, how many cell types it covers, and cancer normal, for cancer type cell types are 278 cell types are there, and how many marker sets are there, and what are the total marker genes are there, like that it is there. Okay, unique genes it covers, and same for other information as well. Okay, what you can do, like you can go to downloads and you can download this Excel file. Like if you're working with human sample, you can download this one, or mouse, like that, or you can combine everything. So I have downloaded the human cell marker Excel file. I will just show you how does it look. Okay, the Excel file is like this. So the first column is like what species, because I have downloaded human, so it will be human data, and these are the different tissue. You can see like abdomen, it covers like bone, blood, many tissues covered. So this way you can filter that for your sample. And this is a different cell ontology term, and cancer type, like what cell types are covered under cancer. And again, here you can see the differentiation, whether it's a normal cell or cancer cell. So yeah, you can filter, use like this, you can use the filter. And this is the main thing, cell names, like what the cell, the types. Okay, and this is the marker gene, for example MERTK. This is the marker gene for macrophage. Similarly, CD, this is the again for macrophage, like FAB4, that is for a brown adipocyte, like this, and give also corresponding gene ID and extra information, which literature, and like that. So if I just select how many are there, around 51 markers, 51,486 such gene lists are there. Each row is one gene or one marker, and it tells like this marker which tissue and which cell type that marker is. So with this information, and whether it's a normal or it's a cancer, so we can filter and we can select the marker genes what we need in our use case. Okay, so if you come back again, they also provide something interesting called cell annotation. This is where what you can do, like you can select a species, let's say human, and you can choose a tissue, let's say or let's say I want to try brain, and maybe with that I want to, these are let's say marker gene. So you can from Seurat, let's say you have done cluster 0 marker, you can select and paste it, comma separated, and you submit it, and it will give in that human and in that tissue how is the expression. So I think this marker, based on that, this CD13 is expressed in pericyte. Okay, and this one is natural killer cell, and this is expressed in all these 3 cells. So like that, based on if you combine everything, the best guess is what it gives, like it says that probably if these 4 are your marker genes in your cluster, then probably this is the mesenchymal stem cell, like that. Okay, so we can try out with this one, let's say this macrophage we can take, and just we can, let's try. Okay, I don't know whether it will be work or not, because I have not tried it before. Okay, so now, but see, of these 4 genes, they have the information for human and brain. Let's do one thing. Okay, let's select abdomen, right, because this is from abdomen. If I do abdomen, so now if I submit, all this 4 marker will come, and because it's from there, right, it is expressed. So let's change it to brain only and submit. You will see that of these 4 genes, these 2 are the marker genes, and this gene is expressed in all these 5 cell types, whereas this one is this one. And from this, based on that, the wild guess is like probably this will be macrophage as well as microglial cell. So this is a very quick way to try it out. This is again not statistical based. Probably I don't know what tool they are using behind the scene. Probably they are using this filtering to get it, but this is one way to quickly glance what annotation will be for you. Okay, and alternately you can go to browse. So now let's say you understood what cell type it is, but furthermore you can also if you want to understand more, you can do like this, like you can cell type, and let's say you are interested in activated T cells. Okay, and you want to know more about it, like what kind of markers in this cell type is there. So you will see a bubble, a word chart kind of thing that will tell like, okay, for this is CD4 marker is there. So let's try activated naive T cell, and for this the 2 marker genes are these 2. So that way, if you have already some information, sometimes see, so you some other tool has given some information. Let's say, okay, you have run some tools, and let's say for a given cluster, it says predicted that it's, let's say, adipocyte. So now you can come back here in this type database and check, like, okay, in adipocyte which genes are important, and whether that genes is present in your top 20-30 genes or not. So there, this kind of information will be useful. So if you are using CellMarker, then again coming back is no use. You can refer other databases to cross check. But if you have used other databases, you can come back and use this information to derive the more confidence. So that is one tool, CellMarker 2.0. Okay, so we'll be in the next we'll be using this database, this data in our practical. In the next video, where we will be using this automated method, right? We'll be using CellMarker is one database. Okay, so next move to the second database, that's that MSigDB. Okay, this has data for both human and mouse, and they have curated from many tissues, And the gene set, basically this is a tool for gene set enrichment analysis, GACA, a platform. So you can use that platform to annotate cell type using the GACA platform. And again, carefully evaluate the output by studying the GACA report and statistics before deciding on particular annotation. Okay, so let's go that. Just type in Google MSigDB, you will come land here, and you can see that there are different signatures are there, and we are focused on C8 cell type signature gene set. Okay, so if you go over here, so you will land over here. So within that, you have 866 gene set. If I just open it into new tab just to show you, so these are the different gene sets. Okay, so each if I download one, this will be this gives more information about what that cell type is all about, that gene set is all about. And I think we can download the gene set from here, TSB metadata. If I download, okay, so it will download it, and we can just open it. So yeah, so for this gene set, these are the marker genes that are available. Say so many marker genes are there, but you get an idea, like, okay. Furthermore, what we can go back and I will back to top. Yeah, so we can further browse as well. No, okay, you can go to search now. Okay, sometimes this it's not working here. Huh? Search, you have to give the email. Otherwise, go to download. Yeah, you can log in and do the further investigation. So, but main thing, like if I come over here, right, C8, you can further details, you can find out what it is, so how they have done it. Yeah, so yes, so they cell type from different databases like that. But in short, you will be able to download entire data from back. If I go back, you can download that the entire data gene symbol, and then you can find the marker and use it. Okay, this is one largest large data set, I would say. The third platform is, this is an important one, Tabula Sapiens, that we can use. So this is actually human cell atlas, this human one. It also provides for the mouse. So this is a different database, Tabula Muris. And if you go to Tabula Sapiens, it contains the transcriptomics data of 28 organs from 24 normal human subjects. So they have taken human subjects of 24 and they have covered all the 28 organs, and the all this from this organ they have collected the cell types and the marker cells, single cell studies they have done. So that's why this is a very useful reference data. We can take our and we can compare against this to transfer the annotation as well. Okay, again, so we can take the full data set, or we can also split into different. So I'll just show you how does it look. This is the database, Tabula Sapiens, and yeah, so you can go with the full dataset launch, or you can go with particularly individual, like endothelial, germline, neuronal, like that. So let's try the immune one. So this may be a little slow because it's running on the browser. Let it load. So yeah, so this I have not tried it, but you can explore your data. Probably this will navigate to their cloud platform, and using that we would be able to do that. We will be able to annotate it. Okay, so if I just open, okay, so here you see this is the UMAP projection of all the cell atlas, and see if I just expand it, right, cell type. So here the B cell, so I'm just not able to, but you can see that it's changing, right? I mean, it's very difficult to understand this plot, what it is. But everything is dumped, right? Not get more information, but I'm interested in these collections. Okay. Okay, so here you can filter. Let's say you are interested in what assay or like what cell type. Okay, so this is the important one, tissue. Okay, so let's say you are interested in blood. Okay, so if I just click blood, so now with this blood you have now 51 data sets are there. Okay, from where they have collected it. So if I just go to this one, so from this data set they have what the data set is like, all human PB sample, and if you download it you will get that this H5 annotation. This is Python, this matrix format you will get, and 4G, so the raw matrix data you will get it. Okay, and then you can take this for comparison. So alternately what we can come here, we can explore the data set, the same thing. Okay, this we have got, but I like the CellGuide. Okay, this is very important one. Like I use this one mostly whenever I need doubt. So here let's say you can search like, suppose you don't know, like let's say you have used CellMarker database, you got some information, so then you want to know more about this. Okay, you just type activated, I want to know more about this, what it is. And so what it will tell, like in this Homo sapiens, and you can also filter and select, let's say in the blood, Homo sapiens and blood, and I am interested in this activated T cell. Okay, so if you can further click it, for example, you can you can do like this, like the open and close you can do. And let's say you are now interested in mature TK cell. Okay, want to know more about it. Okay, so it will tell like what it is. Okay, and what are the computational markers are there, and what are the canonical genes are there, so that's a very interesting thing. What I liked about this is like, so if, because this automated method will give you some cell type. Let's say if you want to know more about it, you can come here and you can just search that cell type, and it will give you more information about the marker genes, and also it will give you the information about the cell type as well. So this is very useful information. At the same time, we can also open the gene expression tab, and it takes time actually. I let it load. So what will happen, it will do those genes, right? So these are the genes, and in those tissue overall expression it will show. It will show in a heat map, and 1 means it's highly expressed and 0 means it is less expressed. So let's do one thing. We can probably filter this one. Okay, 135. Okay, so yeah, so you see this particular marker, right? This one is expressed in 83% of the cell, it got expressed. Okay, this gene, and the gene expression, mean gene expression is this one, and cell type is like it's activated T cells, like that. And yeah, I think, yeah, so this is all activated. Now if in case of blood, the expression is very high. You see, this is towards the blue, right? So it's expressed around, the expression is 4.06, but in adrenal gland this marker is not expressed. Okay, so that way across tissue you can also see how that gene expression is varying. That's what it is collecting from the data from this many organs from 24 individual, right? So you can take a gene and you can compare across the tissue as well. Okay, and yeah, so this is a very nice collection that we can use to do. Okay, so yeah, that's about Tabula Sapiens. The last database is the Azimuth database. This is from the Seurat team, so they have developed it. Okay, so if you go to Azimuth, so they have this hub map, that is the NIH Human Biomolecular Atlas project, HubMap project. So what they have done, like, so they have also done the experiment, and with the collaboration they have prepared reference database so that we can query it. So let's say we have the database for human PBMC, motor cortex, also pancreas, like that. Okay, so you can, depending on the tissue, you can choose and go. So let's say if I go to this human PBMC, you can go to I don't know, like recently this GoTo app is not working for me. Maybe you can go and explore. Probably it will open this UMap projection and it will do that, but it's not working. So what I am interested in, like, you can go to these references, and for that databases, what you can do is like you can find the markers here. So let's say this is the level 1 markers, means broad markers. Okay, so this says that, okay, these are the markers for monocyte and these are the markers for CD4 positive, like this. So it clearly says on this sample PBMC blood samples and how many samples were there and what they did, and these are the markers they got. Okay, for cell 2 it's like even more detail, and for the third is like even more further depth of annotation it will come. Okay, so they provide, and all this. One more thing I liked about this Azimuth is this link. Okay, so what they have given, like let's say they have saying that okay, these are the markers for this gene, so and they have given a navigation to an ontology database. Okay, so this points to ontb doesn't support continue to site. Okay, that's fine. So this is the every cell type has a unique identifier that's called the cell type cell ontology. So and it will give complete information about what that cell type and many more information, and you can also see the hierarchy. So that's very important because as I said in the very previous video that you have T cell means within that you have three categories, right? And sometimes we in the cluster we may see these three groups combined together. So that way either you can split them further or you can use this hierarchy to normalize your cell names. Okay, so this will very helpful for the normalization of the names, because CellMarker database will give some different name, maybe Azimuth will give you different name, and MSig database will give you different name. So here if you come back here, this ontology will be helpful for us to annotate it, right? When it's normalize it. So that's where nowadays AI will be very helpful, because AI if have very easy way to comprehensively understand this language, and that's where they are more superior in terms of annotating, because it's not a keyword based match. They understand the name easily, and that's where combining all this method will be useful. Okay, that's all I think in short I had to show in this video. So what in short we saw, like different databases are there, marker databases, and each marker databases the data source is different, the tissue is different, you may see different organs, different cell types like normal, cancer. So that's where choosing a database is very important part. You choose depending on what kind of samples that you are working with. Okay, so next what we will do, so we understand the databases. So next we will start the actual annotation method in the next video. So now first what we will do, we will start with a very simple simple overlap based, means you take this one, you take the marker, just you compare it. Okay, so very simple, no statistics involved, but that will give you idea like how does it works. And for that approach once we cover this general overlap based, that will give you some idea. We'll move to the using these tools like SC type, cluster mole, EG cell type, and AU cell if time permits. But these three will definitely we will cover in this series. Okay, and each are statistical based, and we will discuss those. Okay, so in the next we will talk about this marker based approach. So what we will do, like we will take the markers from CellMarker, and we will take the marker from another DB. As I said, this database is actually somehow not working. It is giving me forbidden, but luckily I had downloaded earlier the data, so I will be using that to show you. So what we will do, we'll have three marker list. We will take our data, we'll compare against all these three set, and then we will see like we'll come to a consensus approach. Okay, so that's what we will see. And yeah, I think I will see you in the next video. But yeah, so many other stuffs are there. I just wanted to show you what all things I have planned, just to give you a glimpse so that you can stay for the next video. Okay, so thanks for watching, thanks for your time, and I will see you in the next video for this custom overlap based approach. Thank you so much. I will see you. Bye.